Okay, so another worksheet. Now, just as a reminder, you want to try and work out these worksheets before listening to the feedback. The feedback is to correct you where you went wrong, to confirm what you've got right? So that's the best way to use these worksheets. So that's what I would definitely encourage you to do with this worksheet as well. So if you haven't done that, go back to the worksheet and read through activity 1. Activity 2 we won't really do. So activity 2 is a game that is really only applicable for people that are in class together. So in the online class it's a little bit harder to do that. So we'll only do activity 1, but I think it will still be very helpful to you. Okay, so when we look at activity 1 here in this worksheet, we're doing something similar to what we did in our last worksheet. Okay, we're looking at, again, carbohydrate metabolism regulation, which is a complex topic. In our last worksheet, we filled out kind of a flow diagram of what would happen to glycolytic and gluconeogenic flux in response to changing levels of insulin and glucagon, that it changes in response to, of course, changing levels of glucose in the blood. Okay, so we're not going to stray too far from that, but we're going to look at different pathways. In this case, we're going to be looking at glycogen synthesis and glycogen breakdown, and we'll be thinking here about this process in two different tissue types. So we'll do it first in the liver, okay, and then we'll scroll down here and we'll do the next one in skeletal muscle, okay, and we'll have different also conditions in which we do this. So for the liver, we want to look at high blood glucose and we want to look at low blood glucose. What's the liver going to do in response to those in terms of glycogen synthesis and breakdown? And with exercise, we will only do during exercise. We've only really talked about what would happen to glycogen synthesis and breakdown during exercise. Okay, so that's what we'll be doing in that one. All right, so let's go ahead and start then high blood glucose. Okay, these conditions here. And we'll be looking here at these enzymes and their function. Okay, now let's go ahead, before we get to high blood glucose, let's just go ahead and think about the function of these enzymes. And that function will, of course, be the same throughout this entire discussion. That's true both in the liver and also in the skeletal muscle. Okay, so when we think about this, glycogen synthase, if you remember, catalyzes the key regulatory step in glycogen synthesis pathways, and what we can say is that it makes alpha 1,4 bonds. Okay, and this would be alpha 1,4 bonds between glucose molecules. So it essentially builds the glycogen granule. Of course, there's other activities that are needed, but those activities are not regulated. Okay, this activity is regulated. Okay, you need branching enzymes and other things like that. Okay, and UDP pyrophosphorylase and that kind of enzyme, right? But in this case, we only care about the regulatory enzyme, that's glycogen synthase. Okay, now glycogen phosphorylase would be the other enzyme that we want to think about here, and this does the opposite of glycogen synthase, and this breaks alpha 1,4 bonds. Okay, and those are, again, alpha 1,4 bonds that are holding glucose molecules together in the glycogen granule. And so when it's breaking them, then of course glycogen phosphorylase is breaking down glycogen and providing then the needed glucose for the cell, or in the case of the liver, it would be exported out into the tissues, okay, or the bloodstream. Okay, now go one step down. Now, just so we know, by the way, I'm going to just go ahead and use abbreviations. So we'll probably use abbreviation GS. We'll use GP for this abbreviation. And then we have down here phosphorylase kinase. Sometimes it's called glycogen phosphorylase kinase. So those are the same enzymes, phosphorylase kinase or glycogen phosphorylase kinase. So we'll just call it phosphorylase kinase for right now. Okay, but know that that's glycogen phosphorylase kinase. And it does exactly what the name says it does. It's a kinase of glycogen phosphorylase. Okay, and kinases, if you remember, they phosphorylate things. So we can say this. I'll put P with a circle, and P with a circle means that it's phosphorylating something. Okay, and it's phosphorylating GP, glycogen phosphorylase. Okay, and that phosphorylation activates. Okay, so we can just say activates. Right? Not all phosphorylations, as you know, are activating, but in this case, glycogen phosphorylase kinase, when it phosphorylates glycogen phosphorylase, activates it. Okay, and then finally PP1C, which is already an abbreviation, so we'll just leave it at that. We'll just say this D phosphorylates, and we'll just say stuff. Okay, because it has lots of targets. Okay, it's rather complex in terms of the targets that it can participate with, and its regulation is rather complex as well. Okay, so we'll of course be looking at that, and we'll be thinking about, is it dephosphorylating, is it, it's always dephosphorylating, but is it activating or is it deactivating? Okay. Okay, so now let's look at high blood glucose. All right, and in the liver, if glucose in the blood is high, remember insulin signaling is present, and the liver is going to then act to take glucose in, absorb glucose in from the bloodstream, and then use that glucose. Okay, it can use it through glycolysis, as we discussed in our last worksheet, and as we've been discussing in class. And if it does that, then it will make them pyruvate and acetyl-CoA, and those two in the liver can be used to do things, right? So acetyl-CoA, though, and in particular in the presence of insulin signaling, will make lipids. Okay, and then the liver will ship those lipids out, and those will be stored in adipose tissue. Okay. The other thing that the liver will do when it absorbs glucose is it will restore any glycogen that has been broken down in the process of glycogen breakdown when blood glucose was low. Okay, and that's what we're going to, of course, think about is how it's going to be making glycogen. Excuse me. All right, so when we think about this, then in terms of high blood glucose, we would expect the glycogen synthase would certainly not be inhibited, okay, because what the signals in the liver cell are such that glycogen synthesis needs to be upregulated, and glycogen breakdown needs to be downregulated. So if we go to glycogen phosphorylase, okay, in high blood glucose, we could put the same X here, and what these X's indicate is that those enzymes are not being inhibited or activated. In the case of glycogen synthase, inhibited. In the case of glycogen phosphorylase, activated in response to high blood glucose. Okay? All right, now, when we then look at this, right, we have to think about, well, how is the enzyme going to be activated or inactivated? Okay, inhibited. Okay, so in the case of glycogen synthase, how is it activated? Well, glycogen synthase it's inhibited when it's phosphorylated. Okay, glycogen synthase is a rather complex molecule, and in terms of its regulation, and it has multiple phosphorylation sites, and there are multiple kinases, okay, that will phosphorylate it at different sites. And the more phosphorylated it is, the more inhibited it becomes. Okay, so then the opposite of that is true. The more dephosphorylated it is, the more active it becomes. Okay, and so what we see here is that we can just say this, that PP1C is going to be the phosphatase. Okay, it's a dephosphorylate stuff, right? It's going to be the phosphatase that then dephosphorylates glycogen synthase, activating it. Okay, now we'll get to how does PP1C become active, but for now we'll just say that that's what's going to happen. Okay, right now glycogen phosphorylase, okay, this is going to be inhibited by the same enzyme. Okay, so it turns out the glycogen phosphorylase, okay, when it's phosphorylated, it will be activated. Okay, so PP1C will remove any phosphates on glycogen phosphorylase, and when it does that, it activates it. I'm sorry, it doesn't activate it, it inhibits it. Okay, because the phosphate is activating glycogen phosphorylase. Okay, so PP1C is going to then inhibit this. Okay, right now, when we then come down here, okay, let's look at these next two enzymes staying in the same conditions here. phosphorylase kinase and PP1C, we want to think about, well, what would be happening to these? Okay, well, glycogen phosphorylase kinase, as we mentioned, activates glycogen phosphorylase, but we know that glycogen phosphorylase can't be activated. Okay, it's inhibited in this process, so it stands to reason that this would not be activated under high blood glucose. Okay, so we're going to put an X here. Okay, now hopefully you followed that reasoning. If phosphorylase kinase activates glycogen phosphorylase, then if we were to activate phosphorylase kinase, we will also activate glycogen phosphorylase. Okay, but we're saying that glycogen phosphorylase needs to be inhibited in these cases, okay, and not activated. Okay, all right. So what then will happen to PP1C? Well, we notice up here that PP1C has to be activated so that it can activate glycogen synthase and inhibit glycogen phosphorylase. Okay, I know this is the terminology and what's activated and what's inhibited. It becomes a headache, and that's why organizing your thoughts in a table like this can be very useful and very helpful. Okay, but nonetheless, we want to say that, well, we can't inhibit PP1C, because if PP1C is inhibited, okay, then it couldn't carry out the activation of glycogen synthase and the inhibition of glycogen phosphorylase. Okay, so let's think of then to ourselves, well, how does PP1C become activated? Okay, and the activation of this, I'm not going to be able to write all this down. Okay, so I'll kind of sum this up with a comment, but let me go ahead and describe the mechanism. Okay, so PP1C is bound to, okay, the GL scaffolding protein, right? The targeting protein, if you remember. Okay, there's these proteins that are found in glycogen. They're called glycogen targeting proteins, and in the liver, it's called GL. Okay, and it's truly a scaffold. It holds together lots of different proteins. Lots of proteins bind to it, so it's a structural protein. Okay, but PP1C binds to that along with glycogen phosphorylase. Okay, so this enzyme here is bound there, and if glycogen phosphorylase is phosphorylated, okay, then it will inhibit PP1C. Okay, however, in the presence of glucose will bind to glycogen phosphorylase, okay, and when glucose binds to glycogen phosphorylase, it changes conformation and it reveals or exposes phosphate groups on it. Well, PP1C, when those phosphate groups come out because glucose bound to glycogen phosphorylase, PP1C will remove them, okay? When it removes them, it actually is released from glycogen phosphorylase and becomes fully active. Okay, so it's kind of one of those hard things for students. It's like, well, wait a second, you just said that phosphorylated glycogen phosphorylase will inhibit PP1C, but then you say PP1C removes the phosphates even though it's inhibited. Well, yeah, that's what I said. Okay, because that's what we find out is true. Okay, so it turns out that when we think about, again, inhibition, we don't want to think about on and off. We want to think about dimmer switches. Okay, and so when glycogen phosphorylase is phosphorylated, it's dimmed, right? Its activity, PP1C, activity is dimmed. Okay, and then what happens is glucose binds to glycogen phosphorylase, the phosphates come out, and there is a small amount of activity that PP1C has. Well, it's in direct contact with glycogen phosphorylase at this time, and that small amount of activity helps it to remove the phosphates from glycogen phosphorylase. That then allows PP1C to then be released from glycogen phosphorylase and becomes fully active. Okay, so the dimmer switch goes up. Okay, so what we're going to say here, so I have to sum all that up in writing something here, we'll say glucose concentrations go up. Okay, and these would be in the cell. Okay, remember we have high blood glucose here, so the liver starts to absorb glucose that binds to phosphorylated glycogen phosphorylase. Okay, and so then this leads to a conformational change. The glucose goes up, it binds to glycogen phosphorylase, it leads to a conformational change, and glycogen phosphorylase becomes dephosphorylated. Okay, so we'll just say this: GP is dephosphorylated. Okay, and then that leads to the activation of the full activation of PP1C. Okay. Whew. I didn't make this up. Okay, don't blame me. Okay, don't shoot the messenger. All right, this is just what we find is happening in the cell. Okay, now come up here with phosphorylase kinase, and when we look at phosphorylase kinase, it's active when it's phosphorylated, which we haven't really talked about. We'll get there in a little bit, but it's inactive when it's dephosphorylated then. Okay, and so what's going to dephosphorylate it? PP1C. Okay, so you might start thinking to yourself, man, I'll just put PP1C as the answer and I should get something right. Okay, well, in some ways you're not wrong. Okay, PP1C is very important in obviously the activation of glycogen synthesis and the inhibition of glycogen breakdown. Okay, and you'll notice that it's doing reciprocal things in these two pathways. Okay, it's always going to dephosphorylate, but the phosphates that it's removing in some cases activates and in other cases it deactivates. Okay. All right, now let's move over to then low blood glucose. Okay, so for some reason glucose in the cell, or in the blood is going down. Okay, so this means that we're in a fasted state, and what happens there? Okay, well in this case we want to put X's, and we think to ourselves here that the liver needs to, in this case, export, not import, but export glucose. It has to excrete it, okay, which means that it needs to produce it, okay. And then our last worksheet, we looked at how gluconeogenesis would be activated by glucagon signaling. Okay, so glucagon signaling will start to rise in a fasted state and will activate gluconeogenesis. But what we ignored in our last worksheet is the fact that glucagon signaling will also lead to the activity of glycogen breakdown in the inhibition of glycogen synthesis. Okay, so when we think about this, we don't see glycogen synthase being activated in this case, right? Because that would then take glucose and put it into the granule. Well, that's not going to help the liver maintain proper blood glucose levels, okay? Because the liver needs to either make glucose from glycogen or make it from gluconeogenesis and then excrete it, okay, or export it. Okay, now that then, if we come down here, right, glycogen synthase is not going to be activated, it's going to be inhibited. Glycogen phosphorylase is going to be activated and it's not going to be inhibited. Again, we have to break down glycogen. Okay, so we'll remove that. Okay, now, of course, we think to ourselves, well, how does this happen? Well, if you remember, coming back over here, I said, well, glycogen synthase is inhibited when it's phosphorylated and it's activated when it's dephosphorylated. So PP1C is going to dephosphorylate and activate glycogen synthase. So phosphorylation of it must be then inhibiting it. Okay, and that's true. And the signal that is, there's lots of signals, okay, that will do this. But the signal that probably makes the most sense in this class in terms of what we've talked about is pKa. Okay, so glucagon signaling, remember, is going to lead to the active form of pKa. pKa is a kinase, protein kinase A, and it will phosphorylate and inhibit glycogen synthase. Okay, now there are other things that certainly will phosphorylate it and inhibit it, okay, that we could talk about, but we'll just leave it simply as pKa, okay, inhibiting it. Right now, glycogen phosphorylase, if you come over here, I said, well, when it's dephosphorylated, it will be then inactivated, okay, meaning that if it's phosphorylated, it's activated. Well, right, what's going to phosphorylate and activate glycogen phosphorylase? Well, in this case, is phosphorylase kinase. Okay, so this enzyme down here, okay, just below that, this enzyme here. Okay, and that's going to then phosphorylate glycogen phosphorylase. Okay, so remember the name of phosphorylase kinase is glycogen phosphorylase kinase, so it's going to put a phosphate on that, and that's going to activate it. Okay, right now, of course, the next question is, well, you know, what's the regulation? Whoops, sorry about that. What's the regulation then of phosphorylase kinase and PP1C? Right, and certainly we would think to ourselves, and it would be right for us to think that in the case of glycogen phosphorylase kinase, if it's phosphorylating and activating phosphorylase kinase, then under low blood glucose, it can't be inhibited. Okay, so we want to say that that's not going to be inhibited, whereas at high blood glucose, it is going to be inhibited. Okay, right, now, Same thing with PP1C. In this case, remember PP1C, if it's active, it's going to, right, do all this activation and inhibition, activation of glycogen synthesis and inhibition of glycogen breakdown, right? So we want to say that that's not going to be activated. It will be inhibited, okay, during high blood glucose, okay? All right, well, now we need to look at, well, what causes the activation of glycogen phosphorylase kinase? Well, our friend pKa. Okay, so pKa, remember, is activated by glucagon, which is released in response to low blood glucose. Okay, and pKa is going to then phosphorylate and activate phosphorylase kinase, which will then phosphorylate and activate glycogen phosphorylase. Okay, right. Lots of very, very similar words there, I know. Okay, but that's what's going to happen here. Okay, now PP1C, what is going to inhibit PP1C? Well, we already talked about it actually over here. And what inhibits this is a phosphorylated form of the enzyme glycogen phosphorylase. Okay. In this case, glucose levels are not going to be rising in the cell because the liver cell is not going to be taking in glucose. It's going to have to make it and export it. So glucose levels, okay, are actually being exported. So we don't see the binding of glucose to glycogen phosphorylase. Furthermore, we see an enzyme, phosphorylase kinase. Phosphorylase kinase. I underlined the wrong one. Phosphorylase kinase that's activated by pKa that is phosphorylating glycogen phosphorylase, okay? And then that is going to then bind to PP1C, okay? And PP1C will then be inhibited, okay? All right. Now that's then, right, I mean, that's such a headache, I know. I mean, I felt like I was just saying phosphorylase kinase and phosphase and inhibition, right? I mean, it's just terrible, all the words that sound so similar, but that is indeed what we have to think about when we think about these pathways. Okay, now let's go ahead and move to then skeletal muscle, okay, and we're only going to look at this during exercise, okay. The functions that each of these enzymes play, right, are going to be the same as what we talked about in our last slide, so I'm not going to rewrite those out, okay, but I am going to go ahead and write down the abbreviations just so we're all clear on those things, okay. Now, so those functions are the same. They're the same in the skeletal muscle. Okay, we have skeletal muscle though here, and regulation, of course, of these pathways is different because the skeletal muscle plays a different role in our bodies, as obviously you know, relative to the liver. The liver is very important in maintaining proper blood glucose levels, whereas skeletal muscle is really important in movement. Okay, so it's going to use the glucose that it absorbs to make ATP. This is especially true when exercise occurs, okay, and when exercise happens, well, then in that case, we see that there's a great need for ATP and then a great need to break down glucose to pyruvate, and then if this is aerobic conditions, then that pyruvate will be further broken down into CO2 and water, okay, through cellular respiration, which we'll talk about later in the class. Okay, all right, so when we think about this then, okay, so if we're exercising, let's go ahead and put some X's here. If we're exercising, you do not want to be making glycogen, okay, because of course the cell is using glucose for energy needs. So we're not going to activate glycogen synthase, we'll inhibit it, okay. The opposite would be true of, course, glycogen phosphorylase, which breaks down glycogen. We would not want to see that inhibited, but rather activated, okay? So that the glycogen granule or any glycogen that's stored in the skeletal muscle can be broken down, and when it's broken down, then it can be used for energy needs, okay? Now, the question here becomes, well, what, again, activates and inhibits these things, okay? All right, well, again, glycogen synthase, okay, that's one we'll look at first. That is going to be inhibited when it's phosphorylated, okay, just as it was in the liver as well. Okay, so what can inhibit glycogen what can inhibit glycogen synthase by phosphorylating it? Okay, it's a tongue twister. Well, certainly pKa, okay, can do this. All right, now you might think to yourself, oh, glucagon again, huh? No, It's not glucagon, okay? Glucagon is a signal for the liver. It does not bind to any receptor found on the skeletal muscle, okay? So the skeletal muscle is not a target of glucagon signaling. However, epinephrine is, okay? And when you exercise, epinephrine is actually released into your bloodstream. And when that happens, then it can bind to skeletal muscle, okay? And it will activate through various pathways, pKa, and those pathways, of course, we've already talked about, okay, the production of cyclic AMP and that kind of thing, okay, and then when pKa is activated, it can then phosphorylate and inhibit glycogen synthase, right? Now, glycogen synthase, like I said, is complex. There are lots of different kinases that will target it and will phosphorylate it and inhibit it. Another one that is prominent and that I want to go ahead and mention is AMP kinase. Okay, AMP kinase is activated when AMP, okay, the molecule adenosine monophosphate, increases in a cell, right? That, of course, if AMP concentrations are increasing, that's an indication that the ATP is being broken down. Of course, when we exercise, ATP is indeed broken down, okay? That is essentially why we exercise, is that we want to burn calories, okay? And we can do that by burning ATP. Its breakdown product is ADP, but ADP, okay, there's an enzyme that will combine two ADPs with each other to make AMP and additionally ATP molecules. So AMP concentrations will go up in this case. They will activate AMP kinase, which will then phosphorylate and inhibit glycogen synthase. Okay? All right, now come to glycogen phosphorylase, right? That needs to be activated. It needs to be activated so that glycogen can be broken down so that we can continue to burn glucose and continue to exercise. What is going to activate that? Well, PK, phosphorylase kinase, okay? Glycogen phosphorylase is activated when it's phosphorylated, okay? And so it will be phosphorylated by PK or glycogen phosphorylase kinase, just as it is in liver, okay? So it's not much different than what we looked at up here, right here, okay? Now, of course, the question is, okay, well, what about glycogen phosphorylase kinase, and what about PP1C? Well, glycogen phosphorylase kinase, if it's activating glycogen phosphorylase, then it itself needs to be activated, okay? So it's not going to be inhibited, again, in this case, because we need glycogen breakdown to be occurring. So what is then going to activate glycogen phosphorylase kinase? Well, again, pKa will do this, okay? So that's one of the means to do this. It will phosphorylate glycogen phosphorylase kinase and activate it, okay? Now, importantly in the liver, it's also important to recognize that the signal of calcium, okay, will do this as well, okay? So calcium levels are going to rise in the cytosolic environment of a skeletal muscle in response to contraction, okay, muscle contraction. That calcium can actually bind to and activate glycogen phosphorylase kinase, which will then phosphorylate glycogen phosphorylase, activating it, and then, of course, glycogen is going to be broken down, okay? So calcium, when those levels start to rise in a muscle cell, they can play a very important role in activating glycogen breakdown. There's also, if you want to come back up here with glycogen synthase, there's also a kinase that's activated by calcium, okay, that will then inhibit glycogen synthase, okay? It's a more minor point, but it is something that we can talk about. Okay. Now, last thing here. Okay, PP1C. PP1C still does the same thing. It dephosphorylates things. Okay, and what we see here once again is that phosphorylation is either inhibiting glycogen synthase or is activating glycogen breakdown. Okay, so we want to avoid having a phosphatase that's activated that could target these different enzymes and therefore disrupt what essentially the exercise is telling the cells to do. Okay, so this does not need to be activated, so we need to put an X here, and it needs to be inhibited. Now, inhibition of this, okay, again, I'm going to describe it, and I'm certainly not going to write the whole thing down, okay, but what we can say is this, that either AMP kinase, this guy, or pKa is going to is going to phosphorylate the glycogen targeting protein, which goes by GM. okay, in this case. Okay, and so it's going to do that, and when it does that, PP1C, which is normally bound to it, will be released from the glycogen targeting protein. Okay, when the PP1C is released from the glycogen targeting protein, or GM, it will be bound by, quote unquote, the inhibitor protein, a phosphorylated form of the inhibitor protein. And when the inhibitor protein is phosphorylated, it will inhibit PP1C. Right? It's a great name. I mean, it's an inhibitor protein, so it binds things and inhibits things when it's activated. When the inhibitor protein is activated, it inhibits things, which is terrible, right? I mean, this is terrible language, but that's the way we have to say it, okay? And it's only activated, the inhibitor protein is only activated when it's phosphorylated, and it can be phosphorylated by pKa and AMP kinase, okay? So what we're going to do here to try and sum that up is that we're going to say that pKa is activated, that will then phosphorylate the inhibitor protein. Okay, inhibitor protein. A little box around that, and then this is going to inhibit, put a little minus sign here, PP1C. Okay. Great, and I'm just going to leave out the phosphorylation of GM and the fact that PP1C has to be released, but that is indeed what happens. Okay, right now, activity 2 is this taboo game, so it's not really conducive necessarily to the virtual environment, because I can't put you in pairs of 4 and have you play taboo, which is fun. Okay, maybe I'll go ahead and put the taboo cards. Make the taboo cards available to you so that maybe you can find your friends and say, hey, I've got this really great game I want to play before my exam in BC403. Would you play it with me? So maybe that will help. So, okay. All right. Anyway, take care. I hope this recording was very helpful.